let's get down to it. We um, we have the dark reactions to go over. Part of the dark reactions is something called the Calvin cycle. Um, it is a cycle, so its starting point is also its ending point. We're really just going to go over the three main phases um, that occur in the Calvin cycle. It will build sugar, and it's going to use ATP and NADH coming from the light reactions. So it enters in as carbon dioxide, and then, like midway through, it'll make a three carbon sugar. So, in order to get glucose, this process has to occur twice. Um, anyway, there's three phases. The first phase is called carbon fixation. So that's when carbon dioxide comes into the system, and it's going to combine with something called, or there's going to be an enzyme there called Rubisco, and it's going to combine with something called RUBP, which is short for ribulose bisphosphate. Anyway, three carbon dioxides will enter, and they will combine with three of the RUBPs in the presence of Rubisco. It's going to make a six carbon molecule real quick, but then it rearranges into six three carbon molecules. It's thought that Rubisco is the most abundant enzyme on Earth, just because this is, in order to support life on Earth, we need a lot of photosynthesis, and this is responsible for a big portion of photosynthesis. So, anyway, that first step is carbon fixation. Next, what we're going to do is we're going to take those high energy electrons that we got incorporated in with NADPH. We'll take some of the ATPs, we phosphorylate our sugar, we reduce it by stripping those electrons out of NADPH. We get glyceraldehyde 3-phosphate, and then we're going to pull one of them out of there, and then we're going to continue on, but that's the second phase, called reduction, or it's basically the reduction of carbon dioxide, and that's happening right there with the NADPH. That's the reduction. And the last one is the regeneration of RUBP. So we're going to take 5 3-carbon sugars, and then we're going to rearrange them into 3 5-carbon sugars, utilizing some ATP, and then basically we get RUBP and we start all over again. It's important to notice that we're taking electrons that have a lot of energy, and we're reducing the carbon dioxide, or these sugars. We're reducing them, we're phosphorylating them, we pull one out, we're good. Like I said, you do this twice and you can get one glucose. Sometimes, though, plants have some problems, especially if they're in like warm climates. When it becomes really hot out, the plants have these holes in their leaves called stomata, and then their cells kind of to the left and the right of the hole, called guard cells. When it gets hot out, those guard cells close up. And then as a consequence, oxygen, oxygen builds up, and the plant can't get the carbon dioxide it needs to do photosynthesis. All of this is to just kind of prevent water from leaving the plant. Anyway, what happens is that once you get this buildup of oxygen, you undergo something called photorespiration. Photorespiration, you're taking like oxygen, you combine it with some stuff, and you basically burn ATP. Yeah. So, you're utilizing some sort of organic fuel, or it's like a sugar or what have you. But you're not getting any ATP, and that's a problem. And the last thing that the cell wants to do is start preserving, um, or keep itself from dehydrating, but then at the same time start consuming all this like fuel. Um, just doesn't want to happen. So there are two ways to kind of do this. You can kind of separate the carbon fixation and everything else in space and in time. Um, whoops. Anyway, yeah, some plants, almost like 50% of the carbon fixed by the Calvin cycle can get drained. So the plant doesn't really want to do it. That is photorespiration, that is. Anyway, there's a type of grass called C4 grasses or C4 plants, and what they do is they separate up everything in space. They do the carbon fixation in one spot, and then everything else in another spot. Needs an enzyme called phosphoenol like pyruvate carboxylase, or PEP carboxylase, and basically that has like a really high affinity for carbon dioxide, more so than Rubisco. So that's going to able to kind of scavenge up all the carbon dioxides. Anyway, and then when they do, kind of their catalysis, make a 4-carbon sugar. Hence, that's why this is called a C4 process. So carbon dioxide, it'll combine with PEP carboxylase, do a couple of metabolic transformations, and then they move in space into what's called a bundle sheath cell, which is around the vein. So we're going from like the palisade mesophyll into the bundle sheath, and at that point, the carbon dioxide gets released, it goes through the Calvin cycle, and so on. So that's a separation in space. Basically, you carbon fix in one cell and then release the carbon dioxide in another cell. Then we have something called CAM, and this is actually separation in time. So CAM is short for crassulacean acid metabolism. These are your succulent plants, aloe vera, some cactus, and so on. Anyway, so what the CAM plant does is it opens up the stomata at night. Oxygen, carbon dioxide can exchange. During the day, it closes it up. So carbon dioxide, isn't going to leave the plant. At least, I should say, sorry, water is not going to leave the plant. Carbon dioxide won't either, right? Well, the oxygen, but ultimately you'll get a buildup of oxygen that's problematic. So with the CAM, you can see like a pineapple on the right. Basically, at night, carbon dioxide comes in, gets incorporated into CAM. During the day, it gets released out of the CAM. It goes into the Calvin cycle, and during the day, the stomates are closed up. So that separates everything up in time. And I consider like the C4 pathway in the CAM as part of the dark reactions. So if you're trying to figure out quite where do they fit in, that's where they fit in. Okay, well then I'm gonna sign off. That's the dark reactions, the Calvin cycle, and then like C4 and CAM pathways.